Hi guys, welcome back to the channel. In this video I will be covering the carbohydrates topic of the A-level biology curriculum. So let's begin. So the monosaccharides are the monomers of carbohydrates. So the monosaccharides are the small basic units that make up the polymer, which is the carbohydrate. And there are some monosaccharides that you're meant to know, which includes glucose, galactose, and fructose. And two monosaccharides join in a condensation reaction to form glycosidic bonds. So the bonds that join the two monosaccharides, so the bonds between the two monosaccharides, are called glycosidic bonds, and they're formed via a condensation reaction. And a disaccharide is when two monosaccharides are joined together, and it has the formula C12H22O11. Well, that is C6H12O6. So, well, if a disaccharide is double of that, then why isn't it C12H24 and O12? Well, this is because a water molecule is lost in a condensation reaction. That's why we take two hydrogens away from this and one oxygen to form a water molecule. So in this example, you have two alpha glucose joining together. So what happens is the OH group and the H group from the other alpha glucose turns into a water molecule, and that's how a glycosidic bond is formed between the two monosaccharides. Okay, so you're meant to know that a maltose, so maltose is made of two glucose joined in a condensation reaction, and sucrose has one glucose and one fructose, and finally the lactose is made of glucose and galactose. So maltose, sucrose, and lactose are all disaccharides made from two monosaccharides. And a polysaccharide is made of many monosaccharides joined together, so it would be more than two monosaccharides joining to make a polysaccharide. Okay, so the glucose isomers. So you're meant to know of the two isomers of glucose, which is alpha glucose and beta glucose. So alpha glucose has the OH group at the bottom on carbon 1. So if I say this is carbon 1, these are all carbons. As glucose is a pentose sugar, so it has 1, 2, 3, 4, 5 carbons. So we're looking at carbon 1, which is there, and it has the OH group attached to it, and it's at the bottom. Whereas if we're looking at the beta glucose, the OH group is at the top. So comparing this, as you can see, the OH group is at the bottom for alpha glucose and at the top for beta glucose. So the OH groups are inverted. That's what you should be saying in the questions. Okay, so now moving on to the starch and glycogen. So glycogen and starch are both formed by a condensation reaction, and they're both formed from alpha glucose. So it's not beta glucose, it's alpha glucose. So the glucose is stored as starch in plants. So in plants, the excess glucose that's not needed to be used is stored as starch. And there's two forms of starch, and you can have the amylose or the amylopectin. So the amylose, in looking at this, it has 1,4 glycosidic bonds, which is so it's coiled. But looking at the amylopectin, it has 1,4 glycosidic bond as well as 1,6 glycosidic bond. So it has two glycosidic bonds in each carbon. That's why it's more highly branched than this. So than amylose. So the amylopectin is branched, whereas amylose is coiled. So starch, again, it's a alpha glucose, and you're meant to know a bit about the properties of starch. So it's insoluble, so it won't dissolve in water. So this doesn't affect the water potential of cells. So you might not be aware of water potential just yet, but just remember that it won't affect the water potential of cells. So it's also coiled, making it compact. So remembering amylose, so it had those coils. So this makes it compact. So more alpha glucose can be stored within a a smaller area. And branched, it's branched, which leads to a faster breakdown. So remembering amylopectin this time, it had those branches, and this will lead to a faster hydrolysis and or the faster breakdown. And it's large. Starch is a large molecule, so it doesn't leave cell, so it'd be used up in the cell, and it's not wasted. And looking at glycogen this time, and glycogen is used in animals as a glucose storage. So in starch was the storage for plants, glucose storage for plants, but in animals it's glycogen. And again, it's also made from alpha glucose, and it's very heavily branched, so it's more than starch this time. So it has 1,4 and 1,6 glycosidic bonds, which means there's a faster hydrolysis. So if you have a sprinter who needs that energy quick, and the glycogen can be broken down quickly because it has these loads of these glycosidic bonds, which can lead to faster hydrolysis. All right, cellulose. So cellulose, you might be aware, is used in cell walls, and it's made of beta glucose this time. So it's not a alpha glucose, it's beta glucose, and it has long, straight, unbranched chains. So looking at this, as you can see, this is straight, much straighter than the starch and the glycogen, and it only has 1,4 glycosidic bonds. And the structure of it, the alternate glucose is upside down. So looking at this structure, as you'd be able to see that the CH2OH is at the bottom, but in the next one, so in this carbon, this beta glucose, it's upside down. So the CH2OH is at the top, and the next one it would be down again. Next one it would be at the top. So that's why the alternate glucose is upside down in cellulose. And the chains are joined by many hydrogen bonds forming microfibrils. So let's say this was a chain, so this is a chain, and the chains would be joined by hydrogen bonds, which would form a microfibril. So this would be a microfibril, so these would be joined by hydrogen bonds forming a microfibril. And the hydrogen bonds provide strength and make it rigid, so it can withstand changes in water potential. So if there was any changes in the cell, the cell wall would stay as it is, as the hydrogen bonds and give it strength and make it rigid. Okay, so you're required to know of the reducing sugar test, which is the Benedict test. So maltose, lactose, glucose, galactose, and fructose are all reducing sugar that you should be aware of. So how you do that test, you add Benedict solution to the sample, and then you would heat to 95 degrees. So remember, 95 degrees, you're not boiling it, you're not heating it to 100 degrees, you're heating it to a 95 degrees. So a positive result would give a color change from green, yellow, orange, brick red. So the more reducing sugar is present, the further the color change would occur. So if it has really a tiny amount of reducing sugar, then it would have a green color change. It would turn into green solution. But if it has a lot of reducing sugar, then it would have a brick red, form a brick red solution. So this shows the color changes. So if there's no color change, then it would remain blue, which is the Benedict solution color. Okay, now see, so the non-reducing sugar, again, it's Benedict's test. So this, you're required to know sucrose is a non-reducing sugar. And what you do is you add HCl, hydrochloric acid, and then you would boil to hydrolyze the glycosidic bonds. And then you would neutralize the hydrochloric acid with sodium hydrogen carbonate. And then you would do what you would do in a reducing sugar test. Then you would add Benedict solution and heat to 95 degrees. Again, not boiling it, just heating it. And a positive result again from green, yellow, orange, and to brick red. So the more non-reducing sugar present this time, the further the color change would go. And finally, the test for iodine, the starch test. So you add iodine solution, and if starch is present, then a positive result would indicate a color change, so it would turn blue or black, bluey black.